haploid state, again, for the cell that only has these duplicate chromosomes. And they're already down to half the number of chromosomes they started. For the original cell is all of this. The original cell had all 4 chromosomes. Now we have 2 cells that have only 2 chromosomes. And then when we go through meiosis 2, we end up with cells that only have randomly 4 cells. And 2 of them are going to have this chromosome, this chromosome, this chromosome, this chromosome, this chromosome, this chromosome, and this chromosome, and this chromosome. That's because during anaphase 2, what we're doing is we're separating out these chromosomes from each other instead of the following chromosomes. They separate out instead. And so this status of being haploid remains so with here. So these are N, these are N. We started with 2 N because we originally had all 4 of the chromosomes. Here, we look like you count, if you count the chromosomes, you count 4, but they don't become chromosomes again until they become separated. So the primary spermatocyte, when it goes through that meiosis 1, produces 2 secondary spermatocytes. So that's the name of these cells right here. These are called secondary spermatocytes. That's for the process. They're not sperm. And then meiosis 2, we then go through the process again. You think, oh, now we're going to get sperm. Well, not exactly. At the end of this stage, what these are called at this point are spermatids. They're just starting to grow some flagella here. But the problem is they have too much cytoplasm. And so the last step of this process is called spermiogenesis. This is where basically there's no division during the process. It's just the cells become fully mature, and there's nothing more than these chromosomes that are inside it, and nothing else. So this is spermiogenesis. We lose a lot of cytoplasm. I think I have that. So these are for sperm. This is where we lose cytoplasm. I think mostly I always want to give you this as an example of meiosis in action. And just like we would expect, at the end of meiosis, we have 4 possible gametes that can go and then fertilize eggs. Now, the average male produces about 300,000 sperm per minute. This is more of a factoid than anything else. Now, when we compare that, though, to oogenesis, to the production of egg cells in the ovary, you'd expect to see something very similar. Now, we start off very similar. Instead of a spermatocyte, we have a primary oocyte. So, again, as an oogonia, they get rise to these. In the female reproductive system, the oogonia produces these primary oocytes, and they go away. And the female deals with just those primary oocytes for the rest of their life. So we go through meiosis 1, and then right away we see that there's already going to be a difference. We don't get 2 secondary oocytes. Instead, what we get is an oocyte and a polar body. And to show the polar body, right up here is the corner of the overall, well, it looks like it's still connected to the secondary oocyte. And eventually what happens is it gets absorbed and it's never used. The whole point of producing a polar body is to get rid of that excess genetic material. This still happens. It still goes into the haploid status. It says right there it's 10. And it even shows us that the polar body itself is 10. A little bit all the extra chromosomes, 23 pairs, and split up. There's 23 chromosomes in that. There's 23 chromosomes in the secondary oocyte. They're all duplicated chromosomes like this, but they're present in both bodies. However, the oocyte has kept most of the cytoplasm, like most of the organelles, and all that's left in this polar body is just genetic material. So you don't get two oocytes. You don't get two secondary oocytes. In meiosis 2, the polar body, the original polar body, the first polar body produced, apparently doesn't even go through meiosis. What happens is the oocyte will now go through meiosis. And this is it. This is a kick. It only does the second meiosis if the sperm fertilizes the oocyte. Otherwise, it never happens. It goes through a whole other time until it happens. So once it's fertilized, then it finishes meiosis 2. But from this standpoint, one of these is not chosen. One of these gets reabsorbed. It's just being out there and nothing's done with it. And the second one goes on and produces two structures, but one of these is another polar body. So it also gets absorbed. And so finally, the only thing that's left is very briefly this ovum. But because fertilization has already taken place, the sperm nucleus then merges with the egg nucleus and they form into a fertilized zygote and go right into producing baby. So the actual presence of an egg is very brief. Very brief. And we only get one. We get one cell of this. The other polar body doesn't even bother to go through meiosis 2. It just gets reabsorbed. And this new polar body gets reabsorbed, and all of a sudden this last single oocyte, this last egg that is now going to turn into a fertilized zygote and produce a baby. If everything goes right. So not all meiosis, not all meiosis is the same. Meiosis in the male we get 4 sperm spermatocytes, in the female we only get the one oocyte. The question is why? Why would it be different? I mean, why would we want maximum fertility in 4 oocytes for the upper? And it really comes down to practicality. This egg, I should say this fertilized zygote, has to journey from the fallopian tube, the oviduct of the uterus, all the way to the uterus. That may not sound like a great distance, but when you're one cell and you don't have your own circulation, you don't have your regular nutrient supply anymore, and you have to go through several rounds of mitosis to start to grow a baby, which is what it does. It actually implants this several egg structure into the uterus. That's a lot of energy. And the sperm is not bringing anything. All the sperm brings is DNA. So the DNA comes in and fuses with that nucleus to form that fertilized zygote. And it's not bringing any energy with it. And even this mitochondria are left behind. They want to know your maternal background, your maternal inheritance. They will look at your mitochondria because all of your mitochondria and every single one of your cells came from your mother. You have that for your father. And drawings. So, in order to make that long journey before it can finally implant in the uterus and then create a connection with the mother's circulatory system through the placenta, that requires energy to grow that structure of the placenta from embryonic tissue. In order to make all that happen, the egg has to keep all the cytoplasm. It can't share it with equally these other eggs. It has to devote it all to this one effort. That's why you want to get one egg for every primary oocyte. So there are variations on the pattern. This unequal cytoplasmic division is what results in this particular case. But the meiosis still happens because we have to reduce the genetic material down to 23 chromosomes so the baby doesn't have too many chromosomes or we don't get a viable baby. Too many chromosomes does not mean better. And the other thing I really need to talk about before we move on, any questions about chromatogenesis or oogenesis. This is just for humans, so this is human biology, so that's what we're focusing on, the human side of things. I want you to take a look at this image on the right-hand side. This is this condition called non-disjunction. What they're showing, they're focusing on this one pair of chromosomes. And instead of the chromosomes separated like they're supposed to into two separate cells, both the chromosomes went both sides. Remember during meiosis 1, during anaphase 1, we have the homologous chromosomes are all lining up. And then we've got maybe a longer pair here. And these are supposed to go in opposite directions. Right? That's what we're supposed to see happen in meiosis 1, to separate out these homologous chromosomes. But what happens if that spindle fiber doesn't attach right, and one of these instead sneaks over and grabs that one too? You've seen those masters, they're messy business. And they look as neat as what we're drawing up here. So now we have this errant connection, and instead of this going the way it's supposed to, it goes over here with this pair, and we end up with this non-disjunction, so that this cell ends up with only that one chromosome, and this cell ends up with all three. That's a problem. One of the conditions that, so usually what happens is fatal. It results in a non-viable cycle. It is a bigger chromosome, you just can't be missing that much genetic, or you can't have that much extra genetic material that's bigger. But there is an exception, and that is chromosome 21. Chromosome 21, karyotype growth, is a very short little chromosome. It's just too shy of the second chromosome. It looks a lot like an X, actually. When that happens, when someone ends up with an extra chromosome 21, it leads to a condition known as Down syndrome. Maybe you've heard of it. The more official word for it, official name for it is trisomy 21. However, there are instances of Down syndrome that occur that's not caused by trisomy 21. Sometimes there's a break in the chromosome and an extra piece of 21 goes along with the rest of the chromosome. That usually ends up in a less severe condition of Down syndrome, so you may have very high functioning Down syndrome or lower functioning Down syndrome, and that's what they're referring to. The other instance where trisomy is not fatal is when we have it happen with the sex chromosomes, X and Y chromosomes. I'll give you some examples. Monosomy is also fatal unless it's the X chromosome. So one extra, so this other cell over here that ended up missing in the chromosome completely, the cell over here, that also ends up being fatal unless you're talking about the X chromosome. You can't not have both pairs of chromosomes, which is kind of weird. You think, gosh, we have extra chromosomes, don't we have pairs of chromosomes? But it just doesn't work that way. One of the reasons they think that monosomy is not fatal for the X chromosome is because in women, they have two X chromosomes, and in their cells, what generally happens is one of those X chromosomes becomes inactivated, becomes what's called a bar body. If you actually look at the cells and do a karyotype on those chromosomes, typically the second X chromosome looks like it's kind of shrunken up, so it's even more condensed than usual. And so maybe missing an extra X chromosome is not so out of the ordinary. And the condition that someone has when they're missing an X chromosome is called the XO or Turner syndrome. So they have a single X chromosome for each of their cells. That individual, because default setting for the human condition is female, unless you throw a Y chromosome in there with this SRY gene, you end up with a female condition. These individuals are female, but they're sterile. They don't have necessary genetic support to produce what they need in order to have babies. There may be other issues, but that's the most common. And then some other examples of trisomy conditions, Klinefelters, where you have two X's and a Y chromosome. So what do you think that individual is going to be? Well, they're going to be male, but they're going to be sterile. How many X chromosomes leads to sterility for that? What's interesting though is that an extra X for women, poly-X syndrome, where you have 3 X chromosomes instead of the usual 2, doesn't seem to matter so much. You end up with individuals that can actually have children normally. They work out during meiosis. They produce an egg that has just 1 X chromosomes, so they work all that out. And Jacob's syndrome is the same way. Males with an extra Y chromosome, it turns out not such a big deal. Males, they used to say males have extra Y chromosomes are extra aggressive. That's really hard to say what is extra aggressive. Taller, though, they often are taller individuals. The extra Y chromosome gets a little boost there. The image here is showing us an extra chromosome 21, trisomy 21. So I didn't want to do this. This was not really something I needed to do. So I think what we're going to do, now I'm talking about some unusual things that happen with chromosome disjunction. I want to move on to DNA. What genetic inheritance, right? Where are we at, folks? Crazy morning. Not my usual prep time. I thought to think I should switch the order of this topic anyway because we're going to do DNA today in wild, but I want to keep this on target. So we're supposed to be doing genetic inheritance and then DNA. So let's see what we can do. I don't want to throw you guys too far off if you brought your genetic inheritance notes, which I hope you did. Try to keep it short though. So first we're going to start off by defining a couple of terms. Genotype basically refers to an individual's genes, what genes they possess. Gene locus refers to the location of that gene, where it's located on the chromosome. Put that up on the board. Gene locus. Location on the chromosome. So, if I would explain this, I have yet to stumble upon. So a gene, you talk about a gene, people are thinking about a segment of DNA that gives rise to some RNA that then produces a protein, and that protein has some effect on the body. As long as that's simple, the genes give rise to RNA, and the RNA is just an RNA that does the job in the body too, a tRNA that we'll be working with today, or ribosomal RNA. But the idea is the gene is some product that has a job to do. However, it turns out there's different forms of that gene. It's like basically like a pair of genes. You can have a pair of genes. You can have a gene in a black color, gray color, blue color, different colored genes. They're all genes, but they're different colors, different forms of those genes, different textures. And the same thing for genetic inheritance. There's the gene and its product, but then there's different variations in that. So diversity, variation, that's our friend. Now we tend to use the term allele to describe the different forms of the gene. alleles, different forms of a gene. Now then we can maybe come up with some terminology to talk about different forms. How do we describe them? Maybe we're comparing them. So in this particular example, sometimes you can say that one form of gene is dominant over the other. And so one form of the gene is dominant and the other one is referred to as being recessive. In genetics, when they talk about dominant versus recessive, they're trying to do genetic problems where they have a letter to represent it. Or they could use a word, but they have to use letters. And so for dominant, they often will use a capital letter. And for recessive, they'll use lowercase. So that's what we're seeing here. They're showing us an individual has normal pigmentation and they have two capital A's that indicate that they have normal pigmentation. This individual is albino. They don't produce normal skin pigment, they don't produce normal hair pigment, they don't produce normal eye pigment. They also have, you can also see pink in their eyes, like all albino. And they have two lowercase letters represent this. Albinism is actually a little more complex than this, and so there's actually different forms of albinism, but we won't go into details. This individual is also normal, even though they have a capital A, but they also have a little a. But because capital A is dominant over little a, capital A means normal pigment, this individual has normal pigment. We don't see the little a. But if they were to get together with someone else who had the same sort of genetic condition, they might produce a child that's albino, even though they both the parents have normal pigmentation. Let's see why that is in a moment. So these are the simplest types of genetic problems. Again, just dominant and recessive alleles. And then there's some further terminology to describe the condition of individuals and what combination they have. Remember, we all have two pairs of chromosomes for each chromosome. We have two chromosome 1s, two chromosome 2s, two chromosome 3s, two separate chromosomes. And so that means on each chromosome we have the possibility of having a different form of any given gene. We might have the same form. This individual here has the same form of the genes. They're both capital A's. This individual here has two different forms of the gene. It has a capital A and a little a. We just always see the expression of the capital A. So when they have both of the alleles that are the dominant alleles, we call the condition homozygous dominance. And I only put this up here because I want to emphasize this part. So homo means the same. And then the other condition where you have two recessive alleles is called homozygous recessive, because the alleles are the same. They're identical. So the term that we use to describe someone who has one allele or has two different forms of the gene or two different alleles is heterozygous. Because hetero refers to being two different alleles, refers to being different. Just like we use the same prefixes for heterosexual and homosexual. Heterosexual, two different genders that like each other. Homosexual, two individuals of the same gender that like each other. It's really important that you understand that dominant and recessive has nothing to do with the frequency of that allele in a population. Dominant doesn't mean better. Because this example is in front of you, and there is actually some real challenges with being albino in terms of exposure to the sun, UV radiation, the chance for skin cancer. But recessive condition isn't always a disorder. It is always a lack of something. Sometimes it actually is a condition that's normal. It's not a disorder. So the frequency of allele has nothing to do with whether it's recessive or dominant. because it's a recessive allele doesn't mean it's not rare. It's rare. In this case, it is. All it is rare compared to normal pigmentation because it gives it a distinct disadvantage to the individual who possesses it, particularly if they live in an area where there's a lot of sunshine. There has to be a selective factor in evolution that's going to work to decrease the frequency of an allele that gives a potential disadvantage to its individual. And also, something else I really need you to understand is that a lot of times recessive alleles, not always, but a lot of times recessive alleles actually represent the lack of something. Because it's pretty straightforward. The lack of something is an absence of something, so whenever you have something present, it's always going to dominate over that. So imagine this pigmentation thing. This individual doesn't have a gene to produce normal pigment. They can't. This individual has two genes producing normal pigment. They have lots of normal pigment. This individual only has one gene for normal pigmentation. That's enough. They have normal pigmentation because they have one good gene that's producing pigmentation. The lack of that second gene doesn't matter. There are some genes where the alleles cause, and we're going to talk about that, a different relationship, but it's like clear dominant recessive when there's an actual mixture, and we see that this condition is actually different from either this or this. There's a combination effect between the two. But in this case, all you need is one good gene, you have one pigmentation. Now, this individual going to be lighter than that individual, all the data says no. So a lack of something is generally recessive because maybe in the presence of something, it's going to show itself. It's going to dominate over the other. So a lot about genotype. The phenotype is pretty straightforward. Phenotype refers to an individual's physical appearance. It is the expression of your genotype. There is a relationship there. You need to understand that. Your phenotype, how you physically appear, is determined by your genotype. But to show the reason why we need something like both genotype and phenotype, we'll do is turn our attention back to this figure again. These two individuals are identical in terms of their phenotype. Both their phenotypes is normal pigmentation. They're different from this individual's phenotype, which is albino. But these two individuals' genotype is different. This individual is homozygous dominant. This individual is heterozygous. Phenotype, same. Genotype, different. That's why they have both those terms. Questions? Alright, so now we're going to talk about doing some genetic problems. And this is actually preparation for next week's lab. So we do a genetic determinants. It requires you to do a lot of genetic problems. So we start off simple with a one trait cross. So when they start to set up these problems, they're just focusing in on just a single gene. And potentially more than one allele, but it's a single gene. So that's what they're showing. They're showing we're going to focus on freckles. And the recessive condition here is basically little f, let's do capital. Capital F equals freckles. To get one capital F, you're going to have some freckles. Because it's the dominant condition for the big capital. Little f equals no freckles. And both of these individuals at the very top, I don't know if you're familiar with this, but I don't know what it's like sometimes. This little symbol here is supposed to be a spear and shield. That's the shield, that's the spear, and that. I don't make these rules, and that's like hard and fast rules. Not really fond of these terms, these symbols, but they still use them anyway. And this condition means female, and the idea here is this supposed to be a looking glass or a mirror? Anyway, that's female. That's male. I think tell that's male, the little square in there. They're both got no freckles, and they're both homozygous because they're recessive. Now you can be recessive, you can have no freckles as different homozygous. So that means that all of their alleles, no matter what, there's only one choice. Either a little f or a little f. So all their alleles have a little f. little f, a little f. They all have a little f. So all the children are going to have no freckles. That will never change. Down here, we have an individual that has freckles and they're homozygous dominant. And this individual has no freckles, and therefore they must be recessive. They both still only have one choice. They either give a little f or this one gives a capital F. And when those sperm and egg get together, you always end up with children with freckles. This couple will always have kids with freckles. Because of the male. Okay, that's pretty straightforward. This incorporates our understanding of meiosis because we are starting with two gametes that have half the number of chromosomes. We're focusing on just that one chromosome. We call this also a monohybrid cross. Because mono means one. So it's focusing on just one gene. And we're hybridizing two individuals. The children are hybrids. A strange way of looking at things. We have this following tool that we use to do monohybrid cross. We owe to Punnett here, a scientist who came up with this Punnett square. Allows us to investigate hybrid crosses. So now we have two individuals that are heterozygous for the freckles condition. They both have freckles. And these crosses are done, they do these squares in order to focus down on what are the possible alleles that these individuals can give. So you will see, we draw the square, and you see on one side, they got a little square up here, indicate male and female. But I just like drawing off to the side. So they've got the male over here, and the female over here. And what they're showing us is that the female's egg are going to either have a capital F or a little f. She can't give two F's unless they have a non-disjunction, and then we're talking about genetic abnormality. And the male is the same deal. He can only give a capital F or a little f because this is their genotype. This is her genotype. That is an identical genotype. But then they go and have kids. And I want you to understand that the square is only going to tell you one thing. It's going to tell you the probability of producing certain types of genetic combinations. So there's a chance that they're going to produce a child that has freckles and is homozygous dominant. They won't know unless they do a genetic test for it. Which don't know the child has freckles. But then they have another kid who's another chance they're going to produce a kid with freckles, which are heterozygous, who doesn't know it. And then they could also produce another kid with freckles, and then there's a 1 in 4 chance they're going to produce a child that has no freckles. Because they're both heterozygous and there's alleles hiding out the little f. So there's 1 in 4 chances. There's 4 possible combinations. That's what Punnett square does for you. It tells you all the possible combinations of those alleles and the chances of producing the individuals. So we're going to talk about, well, we don't get it. That's a standard Punnett square. I will show you, they do show you this phenotypic ratio here, where they're saying there's 3 individuals that have freckles and only 1 that has no freckles. We're going to do that. 3 individuals have freckles, only 1 that has no freckles. Doesn't look at genotype. You can't tell genotype without specific tools in place. 5/500. The genotype, if we're going to talk about genotypic ratios, is 1 to 2 to 1. You have a 50% chance of producing someone who is heterozygous for freckles. You have a 25% chance of producing someone who is homozygous for freckles. So there's 1 of those, there's 2 of these, and then there's 1 that is homozygous recessive. So the genotypic ratio is 1 to 2 to 1. And when I draw that out, 1 F F 2 F little f 1 little f little f. That's what that's referring to. That's the genotypic ratio. The ratio of the different genotypes. And as you'll be asked to do that in those genetic problems, you need to be familiar with that. So that's what we're talking about. So back to those individuals who have freckles, and we don't know their genotype. And we're curious, we want to know, so we convince them to hook up with someone who has no freckles and have kids. The things we do for science. This would never happen. Redoing fruit fly is not a problem. You can actually do this exactly with fruit fly, with pea plants, no big deal. Humans, that's going to be medical issues. But if it were to happen, and that's what they typically do with humans, they look at family trees, they look at what we call pedigrees, and they look at traits within that family tree to try to figure out if an individual is heterozygous or homozygous based on the children that they have. So if this individual who has freckles marries someone who doesn't have freckles, we know that they're homozygous recessive, and they can only give a little F and a little F. This individual is heterozygous, they give a little F or a capital F. And we make the combination where we put the capital F with little f, capital F with little f. And that's something I didn't really emphasize much. When I was doing this genetic problem, this next one over here, what they're doing, they're modifying cross. And the other square, you notice I'm going to make the mathematical prediction of the genealogy. Just enough to get the work done. You're going to want to do that when you're doing your genetic problems. Make sure you are complete. And so the male is capital F, little f, the female is little f. And all we're doing is we're taking the two possibilities, little f, and you don't even have to do the second one. They did. I don't know why. But there's no reason to do the second one, because that's the only possibility. This individual produces a capital F or a little f, so we put both of them both here and here. And now what we're going to do is we're going to bring the letters across together. So we're going to capital F over to here. So that one went that way. We bring the little f down here. That one went that way. We'll bring this one over here. Now this is going to produce a combination. By the way, whenever you put these letters together, it is tradition, it's common practice to always put that capital letter first, no matter where they came from. As long as the two letters are the same. We're going to do diagonal crosses. I didn't do this other one because why? Because I'm going to get the same combination. I'm going to get this is a little f, right? Capital F, little f, little f, little f. It's exactly the same. There's no real reason to do that second one. You're just making more work for yourself. This tells you everything you know. One, this will tell you that the individual is heterozygous, because if you end up with a child that has no freckles, the only way that happens is if they had a little f. So it's a way to answer the question, is an individual heterozygous dominant or homozygous dominant? And two, it shows us a different ratio where we're actually producing one person with freckles and one person with no freckles. And the genotypic ratio and the phenotypic ratio are identical. So this over here is showing us what happens if the father was homozygous dominant. If they're homozygous dominant, all of the children will have freckles. But again, I want you to understand this is a probability. So here, 50-50 chance you're going to produce someone with no freckles. And imagine flipping a penny. And you flip the penny 4 times. Is it possible to flip the penny 4 times and get 4 heads? Yes, absolutely. So it's possible you flip that penny 4 times and you get kids that are all have freckles. Doesn't change the fact that this individual is heterozygous, but you never knew. This individual, you can flip the penny as often as you want, you're always going to get freckles. So if you flip the penny here 4 times and you get 4 freckles, and you get 4 freckles here, you never know the difference. But chances are, there's a good chance, a 50% chance, that you're going to produce a child with no freckles, and that will instantaneously tell you that the parent was heterozygous. You're thinking, well, what do? You care because it's about freckles. You do care when you're talking about disorders like hemophilia or Huntington's disease. Huntington's disease is dumb, that's not a good example. Tay-Sachs disease. These are good examples where you really do want to know whether you're heterozygous, whether you're a carrier for a gene that might cause problems for the next generation that might actually affect your kids. And even be a fatality. A lot of things nowadays we have genetic tests for, we do molecular tests, we can take your cells, we can run a PCR reaction and see if you've got the gene that's abnormal. they have one for things like Huntington's disease. But this is the old way. This is how they used to figure it out, whether someone was carrying the gene. So they call it a recessive cross because you pick an individual who is recessive and you know they're homozygous, and you're crossing with someone you don't know whether they're heterozygous or homozygous to find out and get an answer. I'm going to change it up early today. We're going to do a little activity. So let's take out a piece of paper. Again, you do not have to have a device to participate in this, but you do need to write down the answers. So again, for your paper, you're going to put down activity, your name. Today is the 20th. Date. Year 5, Friday. Friday Live. You're Friday Live. Yay, Friday Live. If you are participating on your device, take a moment. We'll let you get your QR code. Are we good to go? Yes, no, maybe so. Scream if I'm going too fast. First enter the start. Like the first start. Alright, not bad. We got 10 answers, that's kind of sad, but some are chromatids. Right, duplicate chromosomes contain 2 sister chromatids. Remember to focus on writing those answers down. You write the whole question down, that's on you. DNA is duplicated during the blank phase of the cell cycle. Uh-oh. DNA is duplicated during the S phase of the cell cycle. But most of the labs said S phase, so I thought you guys were going to nail this one. But then again, they're only thank you. Blank acts as an external signal to stimulate cell proliferation. None of them are correct. Now, progesterone is the correct answer. I'm going to fix this question. Progesterone is the correct answer. Progesterone acts as an external signal to stimulate cell proliferation. Blank is important in tissue repair. What are our choices? Blank is important in tissue repair. Mitosis is the correct answer. Mitosis is important in tissue repair. Meiosis only matters for reproduction, producing gametes. In mitosis, the nucleolus disappears during blank. In mitosis, the nucleolus disappears during blank. Prophase. In mitosis, the nucleolus disappears during prophase. It reappears during telophase. Back to 16, so let's make it happen. Tetrad formation and crossing over occurs during blank of meiosis. Tetrad formation and crossing over occurs during blank of meiosis. Prophase 1 is where tetrad formation and crossing over occurs. Oh, you pick prophase again? And he comes out ahead. There we go. Take a 2-minute break. Genetics is about probabilities too. What's the probability of being that particular combination? Science players are a way to calculate probabilities. We'll look at it physically, but really you're talking about a 1 in 2 chance of producing a gamete that has this particular lorem, 1 in 2 of those. As it gets together with another individual, not this individual, but someone else who has capital letter, middle letter, then the chances are 1 in 2 that they're going to produce a gamete that's a little better. and you multiply this across the board. 1 times 1 is 1, 2 times 2 is 4. You have a 1 in 4 chance of producing an individual, which is what we see here. A 1 on a 4 of producing an individual and a freckles. So you can figure that out for like one event occurring using multiplication like that. And then add is determine the probability, sum of probabilities for a given phenotype. So when we look at all of these, there's a 1 in 4 chance that you're going to produce someone with freckles. There's 1 in 4 chance to produce someone that are heterozygous for freckles, or 2 to 4. Another way of looking at that. And basically, you're going to add up those possibilities. So you have 1 in 4 that's homozygous for the freckle condition. And then you have 2 and 4 that is heterozygous for the freckle condition. And since the denominators are the same, then we just add the numerators, and we have a 3 in 4 chance of producing someone that has freckles. It allows us to answer questions about phenotypes with probability. This is a mathematical explanation or something involving fractions, which everybody loves fractions. So a dihybrid cross is where you're looking at 2 different genes at the same time. And that's just something that historically has been done, and so traditionally it's so routine to do this. In this particular combination, you're looking at someone who has freckles, obviously there's a lot of amigos, versus someone who doesn't have freckles, someone who has short fingers, versus someone who has long fingers. And it may surprise you to notice, but short fingers is dominant over long fingers. And because this individual is homozygous for one condition, and this individual is homozygous for one condition, they can only really produce one type of gamete. This one produces a egg as a capital S. This one produces a sperm as a little s. And all of their children will have freckles and short fingers because they're going to be heterozygous for both conditions. A allele for freckles, or no freckles, short fingers, and long fingers. So freckles hasn't changed, we just added a new condition. So we're considering dihybrid crosses now, where di means 2. There's the 2 genes. The new condition, I don't know, I like using test, but that's a good one with. Short fingers is either S or F. Typically, when they pick a letter to represent a given gene, they're going to pick a letter that starts one of the words. But not always, but yeah. This is going to give you short fingers. Little s gives you long fingers. So what happens when you take 2 individuals who are heterozygous for both conditions and you want to do a Punnett square for that? You get a 16 square headache. They're totally doable, but you have to be patient and just sort of work your way slowly through it. So this is talking about 2 individuals that look like this. F, F, S, S, crossed with someone who has freckles and short fingers. So 2 people with freckles and short fingers, but they're both heterozygous for the condition, and then you have to set up a 16 square Punnett square. In order to fill this Punnett square out, you have to figure out what alleles these individuals might produce. And I have a practice that I've always used for this, even though I generally see it right away when I teach, because how I first tackled this way back when I was in high school. This allele is going to combine with that allele, give you my first combination. Then this allele will combine with that allele and give you my second combination. These are all possibilities in terms of the sperm that this individual produces through meiosis. Then we take the second little f. Now here we go with lowercase first because we're using two different letters. If you're really careful with that S, though, make sure you make it really small. That's one of my friends. A little f and a little s. That's this combination right here. So this verifies, this guarantees that I look at all possible combinations. And because this individual has the same genotype, I know that it's the same combination. And then we start putting these together, and you want to practice this because this is what you're going to have to do for genetic problems when we do lab next week. Capital F goes with capital F, they join up. Capital S with capital S. Capital F with capital S. Capital S with lower S. So this individual still has short-shaped freckles. Capital F with lower F. Capital S with capital S. Capital F with lowercase f. And capital S with lowercase s. Fully heterozygous. Capital F with capital F. That's little s. Notice I'm putting the capital letters first, just when I'm putting the two letters of the same type together. Capital F, little s, little s. Somebody who has long fingers. Let's not lose track of them. Capital F, little f, capital F, little s, capital F, little f, and little s, little s. Someone else who has long fingers. Capital F, little f, capital F, capital S, capital F, little s, and capital F up here, little f down here, capital F, little s, little f, little f, capital S, capital S. Somebody who has no freckles. Little f, no freckles. Capital S, little s. Capital F, little f, capital S, little s, capital F, little f, little s, little s. Someone who has long fingers. Little f, capital S, little s. Somebody who has no freckles. Little f, little s, little s, or one individual who's got no freckles and long fingers. Notice I would try to use different symbols in each case to track where the ones that stand out stand out. When I look at that mess, I don't see it. So it's good, important that when you do these is you find a way that allows you to see those unique combinations. Those individuals stand out. So we have three individuals that have freckles, but they have long fingers. We got three individuals that have no freckles, but they have short fingers. And one individual that has no freckles and long fingers. Everybody else here in this party who have freckles and short fingers. All the combinations for gametes from individual heterozygous for both traits. That's what we started with by doing this exercise. We have all the possible combinations for the gametes. These are sperm, I draw eggs, but these are all the eggs. probability for a child with freckles and long fingers. Freckles and long fingers. I've got 3 out of 16 possibilities here. So this math problem that we have here, I want to erase this table. What they're doing is saying, why don't we took these separately? Why don't we just look at each of those separately as this individual cross? So the probability of a child with freckles. So that's the standard cross that we saw earlier. That's capital F, little f, capital F, little f. We make a monohybrid cross like this. We get 2 capital F's. We get capital F, little f. We get capital F, little f. And we get the 1 S and 2 little f's. In other words, a 3 out of 4 chance that I'm going to produce someone that has freckles in that sort of cross. For long fingers is the recessive condition. So I have a capital S, little s, little s. That is going to be short fingers, that is going to be short fingers, that is going to be short fingers. And only one of these is going to have long fingers, so only 1 in 4 chance of producing long fingers. I multiply these genotypes out, or actually in this case it's phenotypes, to determine whether the individual, what's the chances I'm going to produce someone who has both freckles and long fingers. And 3 times 3 is 3, 4 times 4 is 16. And I've just drawn out on the board for you a total of 16 squares and 3 individuals that have freckles and long fingers, 3 out of 16. So the math supports that. This is the phenotypic ratio that you always get when you do a dihybrid cross like this with both individuals that have heterozygous condition. 9 individuals with freckles short fingers, 3 with freckles and long fingers, 3 with no freckles and short fingers, and only 1 that has both recessive conditions of freckles and long fingers. Now, I know that's not going to go over easy. It's why we have a genetics lab where we practice a lot of genetic problems. And even to this day, I've done a lot of these. I still have to sometimes slow myself down and write out the problem in order to get a correct answer. If you rush through them, you create problems for your brain. Maybe you don't. Maybe you see it automatically. I'm not so much that way. I have to work on it. This stuff came pretty naturally to me in high school, but I still have to do the problems. I still came up with a system. And if you're pretty deliberate about your system, you can do these problems pretty easily. So the same question often occurs with these crosses where someone wants to know, someone who's showing both dominant conditions, are they heterozygous or are they homozygous? Is there a hidden allele in there? And you can do the answer the same way. You're basically going to make a cross with someone who is homozygous recessive. So this individual has to be heterozygous for both conditions. This person has got freckles and long fingers. So the only sperm that he can give are the freckles, no freckles are saying, and long fingers. Whereas this individual gives us all 4 possibilities: freckles and short fingers, freckles and long fingers, no freckles and short fingers, and then no freckles and long fingers. And so what you basically expect if this person is going to be heterozygous, then you start to expect to see some individual populations that have basically no freckles and long fingers. And you're going to be in a 1-to-4 ratio again because of the limited possibilities in terms of sperm this individual can get. And it's showing some of the common phenotypic ratios of crosses, monohybrid type. Monohybrid crosses where both individuals are heterozygous with condition, you often get a 3-to-1 dominant recessive. We're going to look at monohybrid crossing of someone who's heterozygous, someone who's homozygous recessive. You get a 1-to-1 ratio, and you get the same sort of situation occurring when you do a dihybrid cross, and you cross someone who's recessive. So it's a way to find out whether they're heterozygous or homozygous dominant for the condition. So predict what genotype the children will have if one parent is homozygous recessive for no freckles and homozygous dominant for short fingers, and the other parent is homozygous dominant for freckles, homozygous recessive for long fingers. Feel free to write down on your activity sheet. Now this meeting of the day, this activity, we're put together again a team, we're putting some ideas, and what our students do in the first week of class, second week, third week of class, all these things. And it's just like a classroom. I don't really want to be there doing that stuff. But I embrace it. I get creative. And I don't care if I'm wrong or right. Because I know the person who's presenting, i.e. me, will love it if anyone just participates. So I went ahead and I said, oh, pick me. So they brought me the microphone and I shared with them all my ideas. And that was great. I mean, they understood how my ideas were great. But they thought it was great that we got to do that. We got to do participation and we got to finish the meeting and kind of out. So when I ask you that, anything you think you put up, and I'm going to think is great. So if you're just willing to participate, you're awesome. If you're not, that's okay too. I'll go on because there's only one person showing me. But, any thoughts? What's that? Freckles and short fingers? Okay. Cool. Exactly. Who wants to elaborate on that though? Who wants to tell me what the genotype is? Nobody? Yeah, so let me show you how to tackle this problem using the technique that I showed you on the other side. I've got two individuals. One parent is homozygous recessive with no freckles, homozygous dog or short fingers. They give us in parentheses their condition. They look like this. Right? They don't tell us whether they're male or female. That's fine. One parent. And the other parent is homozygous dog with freckles, homozygous recessive with long fingers. So they look like this. What gametes does this individual produce? Let's use my fancy technique over there. Little f with capital S. Little f with capital S. Do I need to write it down again? Little f with capital S. Little f with capital S. There are no other possible combinations. That's the only gamete that I get. I do this, I get capital F with little s. Capital F with little s, the same thing. Capital F with little s, it's the same. Capital F with little s. So this is the only possibility I get. Now, in order to make a child, I just need to put these two together. There's capital F, little f, capital F, little s. Freckles and short fingers. So this is the genotype, and that's what they're asking. They're asking about the genotype. So keep that in mind and carefully as the genetic questions when you approach them. This is a reminder that all these processes that we're talking about are the results of meiosis followed by fertilization, putting the two gametes together. So they're showing us an individual here who has a gene for freckles, no freckles, short fingers, and long fingers. And they're showing us the letters here, capital S, lowercase s, capital F, lowercase f. And they're showing us going through meiosis 1. First, they're showing us the possibilities of that, the possibilities of actually what's going to happen with independent assortment. Here, we've got blue and red on one side, red and blue on the other side. Here, we have red and red and blue and blue. That's a possibility. That is how we get genetic variation in gametes. It could go this way, or it could go this way. And when we do this thing here, this exercise, when we do this, that is what we're trying to do. We're trying to take into account both possibilities. Unfortunately, all of these problems are assuming that the alleles of the genes are on separate chromosomes, because it becomes a very different beast when both of the genes are on the same chromosome. Because remember, in meiosis, it's not genes that are migrating to the poles here, it's whole chromosomes. So if the F and S are on the same chromosome, you'd expect very different results. And that's way beyond the scope of this class, so we won't go into that. But if you look at gene locus, you have to look at how far genes are away from each other, what are the possibilities of crossing over, occurring, and swapping those letters. It gets very complex. So here we're always assuming that the genes are on separate chromosomes, and this is showing the results of no crossing over too. This crossing over is not really an issue, it's just showing all the possibilities of the gamete, capital F, capital S, little f, little s, and then we have the other possibilities here, little f, capital S, and capital S, little s. So that's something you don't have to do in order just to do this exercise to figure out all the different combinations. That's a lot more work than this. Alright, so far we've been talking about simple cases of where one individual or one allele is dominant and the other one is recessive. That is not always the case. There is a condition known as incomplete dominance, where the two alleles have equal weight in affecting the phenotype of that individual. And one of the examples here is familial hypercholesterolemia. And when you have incomplete dominance, when neither of the alleles is dominating, then you get a gradation effect. In other words, all three genotypes have a different phenotype. And that's going to be the case for this next example too, but it's even more complex than this. In this case, we're showing that if you have two normal genes, this is where your plasma cholesterol level falls somewhere between a little below 200 and a little below 300. If you're heterozygous for this condition, you have one gene for this hypercholesterolemia, then your cholesterol levels are going to be higher than 300, almost 600. And if you're unfortunate enough to have both genes for this condition, this disorder, then your cholesterol levels go right through the roof, and you end up with this condition where you get these cholesterol deposits actually in the skin. This is usually, if it's not treated fairly early, even then, usually results in a short lifespan. So it's incomplete dominance because the heterozygous condition is sort of this intermediate between the homozygous normal and the homozygous disorder. Or can you say recessive and dominant here because it's not, there's no recessive, there's no dominant. There's just a homozygous, in this case normal and homozygous disorder. The part of the blood type, I think one thing I can do for you on the board if you're thinking about this condition. We can't both erase my 6 times squared. Definitely need it. So, in other words, they're saying that we're picking a letter. Do we use the same letter? Do we use an H for this, and then we use a lowercase h? And this is the disorder. And this is normal. So we end up with individuals who are homozygous or normal, then they have their blood cholesterol levels being in that sort of 200 to 300 range. And we have someone who's heterozygous for the condition, then you have 300 to almost 300, 500, somewhere in that range. And then if you have someone who has the disorder full blown, then you have basically greater than 600 milligrams per deciliter, something like that, of cholesterol in your blood levels. Very, very high cholesterol levels in your disorder. But again, not really recessive, not really dominant, just a convenient way of representing that. Codominance is a little different, and example of this is blood type. I think of another incomplete dominance, and that's flower color. So that's nothing to do with humans, but there's a gene for white, there's a gene for red, and when you white and red together, you get pink. So that's another example of incomplete dominance. But codominance would be where you put red and white together, you get both red and white appearing. And in this case, codominance or blood type, it basically means the two alleles together, you end up with both of the alleles representing each other. Blood type is kind of funky. In order to do this in a way that makes some sense, they use the letter I. They use the capital I and the allele A for what we call A blood type. And then they use capital I and letter B for equal B blood type. And then they use the lowercase I to equal O blood type. And this goes back to what I was saying earlier, the lack of something is always recessive. O blood type means the lack of a particular antigen on the surface of red blood cells. A and B are specific types of antigens. So remember, we all have the possibility of two alleles of any given gene, two different forms of that gene, or two of the same forms. So in order to get the O blood type, the only individual with O blood type is if they have two of the lowercase i's, and this person actually has O blood type. They have to be recessive. And if you have someone who's going to have a blood type, there's two possibilities. It could be I, or they could be I, capital A, little I. Because the presence of A would dominate over the lack of something. And so even that individual is a blood type of A. And individuals who are going to be B blood type often will be IB, or they'll be IB, little I. And the B dominant. So how do we get A and B together? What happens if an individual has a situation where their genes are like this? They've got a capital IA and a capital IB. What's their blood type going to be? Exactly, the blood type is AB. That's how we get AB blood types. That is the co-dominant condition. Both of the alleles are demonstrating they're being expressed in a way that we can detect them. Multiple alleles A, B, and O, but you can only have two. That's my other point. There's three different forms of this allele, but you only get to pick two. By the way, this is when you get individuals like this, I, A, I, B, with little I's, when they get together, and they have children, and there's a child who has a blood type of O, and that child grows up and says, wait, you can't be my parents, your blood type is A, your blood type is B. And they can't, because there's the possibility to have the recessive. In fact, that answers the question, and they now know they're both heterozygous because they have a child with blood type of O. That's what we're seeing in this image here. This is showing two individuals, blood type I, blood type A, and blood type B. The mother's A, she gives either an egg with an A or an egg with an O. Father's B, gives an egg with a B blood type or an egg with a sperm, sperm with a B blood type or a sperm with an O blood type. And then you put them together, this brings A B blood type. Someone has B blood type, someone has A blood type, and then someone has O blood type. So this combination, if your blood type is B and your partner's blood type is A, and you're both heterozygous for O, then you have the possibility of producing all the different blood types. So this makes up the AB combination. Let's just not address the allocation factor. Well, if your blood type is AB, you know what your genotype is. If your blood type is O, you know what your genotype is. It's only if your blood type is A or your blood type is B that you don't know if you're heterozygous or homozygous until you have kids. And even then, you never have a blood type phenotype. So blood type is what I call codominance exemplified. It's the perfect example of codominance. I don't have any other good ones. The other thing I need to prepare you for doing genetic problems before I move forward. Any questions? It's going on, it's talking about sex-linked traits. Sex-linked traits are genes that we find on the X chromosome. I just finished telling you how we didn't really worry about when genes were found on the same chromosome. The one exception to this in terms of when they were doing initial genetic process was the X chromosome. Because effectively, We are talking about two genes on the same chromosome. We're talking about a gene, for this case, for eye pigmentation or an pigmentation, a certain type of cone that detects pigmentation. And of course, a gene for being female. But it's complicated because X always gets together with Y in the case of male, and the Y chromosome doesn't carry anything but maleness on it. So it can't have a gene that has to do with anything in terms of body features, body traits. Whereas the X chromosome is a big chromosome, it has a bunch of other things on it, including the gene for coding for the cone that allows us to determine the difference between red and green. And some individuals have a defective gene on their X chromosome. And if that's the only gene that gets expressed, then they end up being colorblind for red and green. You can't distinguish between red and green very well. And they pass that on to their children, they can also have the same situation. But I want you to remember this when you're doing your genetic problems. Never, ever, ever, ever put an allele on the Y chromosome. The Y chromosome never carries anything. Don't do it. I'm making an emphasis of this because every semester, someone always does it, usually more than one. So it's a real common problem. There is no such thing as a Y-linked gene. Do not put alleles on it. So most disorders are actually, I put this statement down, I must have read it off the book. I'm left it out, so I'm staying more on this side because it's dark over there. I'm having black. But I'm thinking if I can use here, we can see very well. So what does this mean here? I'm going to read through all of this. Let's put what they have before. What does this mean? Let's think about it. Let's try to take this problem apart. Alright, so the chromosomes are just to tell us the gender of the individual. So we don't have to draw the sphere symbol and the mirror anymore. XY is male. Right? XY is male, XX is female. That's all it's telling us. But the reason that we have these uppercase letters on them is because it's telling us that the X chromosome is carrying a little extra something. Now they're just focused on one gene here. There are other genes we're going to talk about in a minute that are X-linked. But this one indicates normal vision. And this one right here indicates colorblind, red and green colorblind. There are other different types of colorblindness, but this is the most common. And it's most common in males. Why is that? It's simply because males only have a single X chromosome. Female has two X chromosomes, so there's always the possibility that the one chromosome has a defective gene, the other chromosome will have a functional gene. So women in general suffer less than this. There are fewer women who get disorders that are X-linked than men because they have this protective chromosome. In fact, they often describe it this way. So here we're looking at a male who has normal vision, they have capital V on their X. And they have a female who has normal vision, but she has an allele, one of her chromosomes has a defective gene on it. Now, remember, XX is female, and both females here in our probability square both have normal vision. Even this one, because they have the capital V that they got from their father, they call the father protective in this manner. So giving a gene that's normal, even though fathers provided a gene that is not normal. But in the case of the boys, the only thing that the father can give is a Y chromosome. The father doesn't give a Y chromosome, you don't get a boy. So this individual, this boy has normal vision, because they got the chromosome from mom and has normal vision. She's being protected. But this boy is colorblind because he got Y from dad and he got the X chromosome with the lowercase b, the colorblind gene from his mom. And that's how mother would find out that she's a carrier for red and green colorblindness. you'll be asked to set up some genes like that. There's some other X-linked disorders. Duchenne muscular dystrophy, the lack of protein, the protein dystrophin, leads to calcium leaks and muscle cell degradation and deposition of scar tissue and eventually death. This is more common in boys than in girls because they don't have that second X chromosome. Fragile X syndrome, which often leads to autism, is more common in boys. The fragile X syndrome, autism occurs more frequently because there are other types of autism caused by other things, but this particular type of autism is more common in boys. And it results from a repeat of what's called a CDG nucleotide sequence. And then hemophilia is a famous X-linked disorder that was discovered in the royal family of England, tracing all the way back to Victoria, who was a carrier for it, and Prince Albert. Prince Leopold was hemophilia issue of nose. Basically, your blood doesn't clot properly, so if you get any sort of break or cut, you bleed, and it's hard to stop the bleeding because you're not clotting properly. And then the crazy, every yellow square is someone who has hemophilia. Squares equals boys, and circles equals females. How many yellow circles are there? There's no yellow circles. Okay, so that tells you just how preponderous the X factor is in terms of sexual chromosomes in the population. I'm not going to ask you to remember the difference between factor 8, factor 9, and hemophilia A versus hemophilia B. So they are both X-linked. So what I'm going to go into now is I want to just make sure that I cover what are called pedigrees so that you will have a chance to answer these pedigree questions when you do that question. So that means I'm going to jump around just a little bit. So first I'll talk about recessive pattern. This is something worth talking about. So basically, when we're looking at patterns, a dominant disorder is when you have two individuals. If you see a pattern where two individuals are affected by the disorder and they produce a child that's normal, then that's a dominant disorder. Heterozygous parents with disorder. So in other words, if you think about it this way, capital D equals disorder, lowercase d equals normal. And so if you have two parents that have the disorder, but they're like this, right? We get the standard monohybrid cross, disorder, 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 normal, right? So it's lowercase when the disorder is, or when the condition for normal is lowercase, then not only that becomes the recessive allele is going to pop up less frequently. Individuals have the disorder can have normal children in those circumstances. Huntington's disease is an example of this. Huntington's disease is a neurological disorder that is usually fatal for the individual in various fatalities. Medical care is really helping improve the outcome for people with Huntington's disease, but it's something that usually starts afflicting a person in their mid-30s. And that means that they don't find out until after they have children, often. Now there's a genetic test for Huntington's disease, along with genetic counseling. The people start wondering whether they should get the test before they have kids. Recessive disorders, let's flip it, all right? Stay with me. We'll get rid of these, and I'll make capital D normal. I'll make little d disorder. So this is a recessive disorder. Then the pattern is that you have basically two individuals who are normal, and they produce one child that has a disorder. So they're what they're called carriers. They're carrier in the heterozygous condition. They carry the allele for the disorder. Parents are normal, but the disorder pops up in children. So, like I showed you before with the hemophilia, we looked for a pattern where there was mostly male in the disorder, and that told us that was actually disorder. If you see a condition where the disorder tends to pop up and is present when the two parents have the disorder, so it's normal, that tells you the dominant disorder. If you see cases where you have two individuals who are normal and have a child with disorder, that tends to tell you that it's a recessive disorder. Those are the kind of patterns that they look for in a pedigree chart. So here, looking at two individuals who are normal that end up with children that have a disorder, that tells them that this is a recessive disorder, it's a recessive pattern. Whereas the dominant pattern, you have at the top here, two individuals who have the disorder, and then you have several children that are normal. Someone has a disorder, gets together, and someone who is normal, and they have children with disorder, and someone is normal. This tells you that you have a dominant disorder. The disorder condition is dominant. All right, let's go ahead and take another break, and then we'll wrap this up and get into some DNA talk. Women with disorder, however, will always have sons with a disorder. Because in order to have disorder, she has to have two recessive alleles. I'm going to talk about an example for autosomal recessive. Most people don't want to think about autosomal means. Remember that chromosomes 1 through 22, 1 through 22 are autosomal chromosomes. We have 23 pairs of chromosomes, so chromosome 23 is our sex gene. So, an autosomal recessive disorder is basically what we're looking at here. Two individuals don't have the disorder, but they are carriers for it, and they end up having someone who has the disorder. So they don't know that they have it. Also with chromosome 1 through 22. The side sac is just such a condition. This is a disorder that popped up in this particular group of people. It doesn't mean that it hasn't been in other groups since then. The gene that is being affected here is one that's responsible for metabolizing lipids, and so the lysosomes don't have that proper enzyme to clear out lipids, and the lipids start to build up inside of the neurons and the nervous tissue of the child. This is usually done before age 3. So the brain cells build up lipids and lead to big development. So again, you have two parents who don't know they have this condition, and then they have a child who ends up with a condition and dies. So it's pretty heartbreaking. That's why they want genetic testing for these disorders, so that people can make informed decisions. I'm not going to go over the rest of the autosomal disorders. I just want to give you at least one example, and then we'll move on to DNA. But this will hopefully help with today's lab a little bit. We'll do that. So we'll talk a little bit about DNA biology and a little bit about DNA technology. It's come a long ways even since I started working. So some review first, DNA structure. Remember that DNA is made up of nucleotides, and all nucleotides have phosphate, they have a sugar, and they have a pyrimidine, they have a base, either a pyrimidine base or a purine base. And you can usually remember the purines from the primitives because of this sort of mnemonic I'll show you. So I can do it. You see the pyramids. U is for you, your uracil. C is for C, your cytosine. The is for T, or thymine. Those are the three nucleotides that are pyrimidines, that are pyramids. The other two by default, A and C, are purines. Those are A and D are purines. I don't have any molecules. Here, if you know that, then by elimination, these must be purines. So this is showing both a pyrimidine-based thymine and a purine-based possibility. And it's also that the sugar for DNA is deoxyribose, showing us the position of the two strands of their condition is anti-parallel, where this one's facing up and this one is facing down. We'll get more into that. Also, this image is good. Just go away. Beta nucleotides. Let's go back here briefly. We're going to look at that next image. I want you to take a look at the sugar that they're showing you here. What they've done is they've numbered the carbons on that sugar. This is the 1' carbon, that is the carbon that's attached to the base. 2', this is the 3' carbon that has an OH group on it. 4' carbon is here. The 5th carbon is taken off of the ring and attaches to the phosphate. So in this case, the 5' is up. And over here, the 5' is down. 3' is up. That is the nature of DNA structure. The two strands, which is what we're going to see in the next image. The two strands are parallel to each other, but they're in the opposite orientation. First of all, they form this double helix. It's a helical structure and then it twists about the sides. It has to do that or make the connection between the bases. Notice here that the 3' ends up here, 5' ends, the 3' ends up here, 5' ends up here, 5' ends down, 3' end is up. And the 2 S's upside down on the phosphates. And then in this case, they made the bases upside down to emphasize their relationship to each other. They're parallel to each other. They run in lines side by side, but they're called anti-parallel because they're in opposite orientation. And Watson and Crick, James Watson and Francis Crick, came up with this arrangement along with the help of the data from Rosalind Franklin, who did x-ray crystallography on it and provided them the necessary data. They figured out that this was the only way that DNA could come together to form this double helical structure. They knew that it formed two strands, they just didn't know how until they realized that one was upside down together. So it would work. And explain all the data that they have. That DNA has a sugar phosphate backbone. This is basically a fancy way of saying that this is what holds these strands together covalently. Alright, sugar, phosphate, sugar, phosphate, sugar, phosphate. They are holding together these lines with the covalent bonds. We talked about chemistry in this class. Over here, same deal. Phosphate, sugar, all covalently together. The two strands are attracted to each other and held together fairly weakly by hydrogen bonds. So these dotted lines mean these are weak hydrogen bonds between partially charged positive charge and partially charged negative charge. You know the oxygen, sometimes nitrogen. The positive charge is always a proton. This attraction is so weak that you can melt DNA. You can make these two strands come apart by raising the temperature up to a little over, right around boiling, sometimes up to boiling. And we make use of that to do some molecular DNA techniques that I might talk about later on. Also, the thing about the DNA structure is that these pairs come together in a specific way. That only A will recognize T and form hydrogen bonds with it. In other words, they have the correct atoms here to be attracted to each other. And G and C are attracted to each other also because, again, they have the appropriate elements here that have the opposite charges that create hydrogen bonds. G and A and T and C, they don't have that. There's something else that's going to impinge their ability to get together. And even T and G don't bond very well together because of the lack of the proton hydrogen bonds. So it drives what they call complement-based pairing. A and T always matches up with each other. G and C always match up with each other. And when you're not drawing it that fancy way, a lot of times when we draw these two together, we'll draw it like this. G forms 3 bonds with C, A forms 2 bonds with T. Actually, I could reverse this. Give you a different way of looking at it. C here, G here, a T here, and A here. That has implications for DNA structure. Areas where there's a lot of A's and T's tend to be weaker than areas where there's G's and C's, but we don't want to worry about that. The other thing that drives this complementary base pairing that I think is important, because we're going to talk about how DNA replicates itself, but it does it based on this complementary base pairing. And that A always recognizes T, G always binds up with C. They're like couples, they always get together. And part of the reason for that is the hydrogen bonds. That's a really important part of it. The other thing that drives this, or allows the cell to see when mistakes have been made, is always across the structure there are 3 rings. 1, 2, 3. 1, 2, 3. You don't get G combining with A because then you get 1, 2, 3, 4, and that creates a bulge in the backbone that the cell recognizes and clips out and reflects there. You don't get T and C getting together because that's like 1, 2, and you've got this gap that causes the DNA to buckle inwards, and the cell will recognize that physicality and it will remove it and repair it. The real danger is T getting together G and A getting together C. They form 3 rings, but they don't have the right hydrogen bonds. So usually we don't produce mutations in our DNA. Usually it's possible, but usually it doesn't happen because of this very efficient way to make sure that only those nucleotides recognize each other. As we talk about DNA replication, it is absolutely critical that you understand that one concept. Before we go any further, I'd like you to add to your activity sheet the following. Okay, I want to give you some time to think about that, and if I got you to think about it, good. And we are going to move on now. We got a little bit of time. Talk about DNA replication. So bring your notes back out. They're making new DNA. Remember, we need to make new DNA, so we need to make new cells. So we're talking about mitosis and meiosis. We're talking about taking a chromosome from the unduplicated state to the duplicated state. So chromosomes start off unduplicated, like so, and we want to take them and we want to make them duplicated. And this is the process that we will use to do so. Okay. DNA replication, making new DNA. So I had a chance to practice that in lab today. Making chromosomes half old and half new. I want you to notice at the top here. So we're zooming in on the chromosome. We're looking really closely at it until we get down to the structure of just the DNA. We're leaving the proteins out of the picture right now. And what I've done is show the double helix that we're just finished looking at here with completely complementary base pairing, and we've split that chromosome in two. We call this a replication fork. And where we've opened it up, now each of the strands from the original chromosome is serving as a template for new DNA structure. So we see A and T, G and C, G and C. Here's an open G, and here's a C that comes floating in and says, oh, you're my partner. Here's a C that says, oh, you're your partner. So we'll understand that the cells have a ready supply of A and T and G and C in their cytoplasm whenever they need to do this process. If they don't have that, they can't duplicate their chromosomes, they can't do mitosis, they can't do meiosis. They have a real problem on their hands if they have to do any of that. If they don't, then they don't care, and that's why they keep their nucleotides. Over here, notice that they're adding these new nucleotides down here, and there's going to be some implications of that as we probably into it. But right now, I just want you to get your head wrapped around the idea. Pull the old chromosome apart, use each of the old strands to serve as templates to make new strands, and then at the end of the process, we'll end up with two chromosomes. Each one is half new and half old. That is what it means by semi-conservative replication. Each chromosome is half the old, half the new DNA that's being constructed. All right, here's the process. So they turned it sideways on you. There's the original DNA chromosome over here, all in a double helix. And we're going to talk about some of the players here that get this job done. And the first one is helicase. Helicase is a little blue ball, and its job is very simple. It needs to unwind the DNA double helix. So helicase. Unwinds the DNA helix. That's where it gets its name from. Gets it spread out. It opens up the replication fork. We have to do that when we're bringing new nucleotides. Nucleotides then line up with their complements. Once you open things up, that's what these are representing. That basically we're bringing in some new nucleotides that are recognizing the A, the G, and the C. Here's the whole sequence. Here's the bases. And then we can put some bases in there. And here, as soon as those are opened up, then we're going to bring in, and automatically what happens is 5 prime, and a T comes in here, and then another T says, oh, you look good, there's A, and C is going to come in here, And over here, what's happening is the T is going to come in here, and A is going to go in here, the C is going to go in here. They come in and they recognize the hydrogen bonds, they match it with a partner, but they have this backbone that's not linked up yet. That's the job of DNA polymerase. DNA polymerase comes in here and they show it has this big brown ball right here, and it will actually start adding nucleotides. However, there's a catch. DNA polymerase cannot start de novo. It can't just say, oh, there's some unattached nucleotides here, I'm going to attach them. It has to have a primer. And so what happens is we bring in another enzyme in here. What I'm going to use. Primase. And primase will start making an RNA copy, except for it's RNA, so we can't use thymine. We have to use, we're required to remember the last steps, uracil. Then it uses adenine, and then use cytosine, and that links them all together. Then DNA polymerase come in here. DNA fold 3. There's actually a 1 and 2 DNA polymerase. And it says, oh, there's a C there, I can add a G. A G comes in, I can link that to this. A C comes in here, I can link that to this. And then when it's going to basically move in this direction, and add a G to the next one. Meanwhile, over here, same deal. Primase had to come in first. And it had to find RNA, or actually it doesn't use 2 U's, and a C. So then we can bring in DNA polymerase. DNA fold 3. And it says, okay, I can add a G here, C here, and it keeps working its way down. DNA polymerase 1 will actually come in here after primase has gone away. And the beta RNA primer is done, and in comes DNA fold 1. Polymerase 1. It will recognize this RNA and it will erase it. And then we'll come in and go bring DNA. Problem is, it doesn't have to do that. Neither one of these polymerases is going to do that. They can't put them together. They'll do the same thing somewhere else. This strand here is often called the leading strand. This is called the lagging strand. And there's a reason for it. And it has to do with the fact that this strand here, the DNA polymerase, once it gets started, this DNA polymerase, not DNA fold 1, once it gets started, it actually can just go ahead and follow helicases. Remember, around all of this is helicases. Helicases is popping this open, and as it gets to here, it's going to pop these open. DNA polymerase 1 is just going to keep on going, because this is the 3' end. It just is going to add new nucleotides to the 3' end. Over here, earlier on, when this first opened up, DNA fold got going in here, and actually already made some DNA here, A, G here, C. It's already got this in place, and so when the DNA polymerase comes up to the sequence that's already been built, it has to pop off. And then this helicase opens up this more and it comes back in around here along with primates to make more copies and start all over again. In other words, the lagging strand moves away, in the lagging strand, polymerase moves away from helicase. And the lead strand polymerase follows helicase. And by doing so, I can just keep on following and keep on synthesizing DNA. Because this is moving away, when the DNA strand opens up more here, it has to pop off and go here and start synthesis again. And every time it does that, it slows everything down. It can't just keep following the helicase and keep adding more DNA, it's moving away from it. And that all has to do with this orientation of 5' to 3'. Over here, there's 5', and this is the 3' end. The DNA polymerase has to go this way. Over here, the DNA polymerase has to go this way, because it's always going to have to that 3' end. And they're trying to show that here without all the A's and T's and G's and C's. This DNA polymerase is following this helicase. It is opening up the DNA here, and it just keeps synthesizing more and more DNA, and just keeps adding to it. Over here, previously this was opened up, and the DNA polymerase came in here and created, you can see actually a piece of new DNA. You can see the RNA primer. It created a fragment here, and then it had to pop off and then come over here and synthesize that and create a fragment there. And here is freshly opened up the RNA primer as it put here, and the DNA polymerase has popped in. And it's making a new DNA, but look what's going to happen. It's going to hit this, and it's not going to be able to do anything. It's going to come to an end, it's going to come off. Meanwhile, this moves down farther, so it'll come off, and it'll come back over here after this opens up and starts synthesizing again and create another fragment here. These fragments are named after the Japanese researcher who discovered them, called Okazaki. And Okazaki fragments, they went and they looked for them, and they actually had a very famous experiment where they interrupted DNA synthesis, and they ran the DNA out on an electrophoresis gel where they could see a lot of it, and they saw these little fragments, which proved his hypothesis, which stands today as an actual major theory of DNA replication. So Okazaki fragments are only present on the lagging strand. They're not on the lead strand, because the lead strand is continuously making DNA without interruption. The lagging strand, it keeps having to interrupt itself to start the whole process again and again and again. Now eventually you're going to go, well, what are you going to do with those fragments? How are you going to fix those? That's where our last enzyme comes into play. So primase made an RNA fragment. DNA fold 3 is the major DNA synthesis enzyme. This is the one that has new nucleotides to the 3' end of the DNA strand. Ligase, well, I've got on this. Ligase fixes these gaps. Remember, it does this gap here because there's a gap because of the action of DNA pull to replace that primer, and it cannot seal this. Ligase uses ATP and the energy from it to fix that gap. Ligase gets rid of the nicks in the DNA. It fixes the Okazaki fragments too, because you have lots of them from the action before, and it's going to link those pieces of DNA together. Ligase glues all the places that are not glued together back together. It takes energy, but it has to be done. Ligase seals the nicks. Mistakes are rare because of this amazing efficiency of complementary base pairing. A recognizes T and G always recognizes C. What's more, DNA polymerase 1 has editing capabilities. It actually can recognize when G has been matched up to T instead, and it can fix that. So mutations are relatively rare in DNA replication for this reason. Okay. What about RNA? I know, that's a lot. That slide is a lot. There's no really easy way to explain it. I've simplified as much as I can. I'm not going to talk about total boundaries or some of the other enzymes that have a role in this. But this is like the bare minimum we need to understand how DNA makes a copy of itself. Any questions? You absolutely have to spend time with this material outside of class. I know many of you are, but some of you need to spend more time based on what the exam scores are. So take that in mind. Now, I'm going to allow you to have notes on the next lecture exam. Alright? So we'll see if that helps you get that 1 page of notes, and I'll be giving you the guidelines for that 1 page of notes, but it's not really 5x4 list right now because we're not past the next lecture exam. So I'll get that along with the next practice exam. Alright, so RNA structure. We're going to compare it with DNA. First of all, there's no double strand. It's all single strand. That's weird because there's some RNAs that will loop on itself and create double strand. We also see here it's a single strand. We can see that it uses the sugar ribose instead of deoxyribose. And of course, as we've already emphasized, it uses uracil thymine. So every time you're tempted to put a T across from an A when you're doing RNA, remember it should be U. And of course, it has that ribose sugar. They're both made of both nucleic acids, both made of nucleotides, both have a sugar phosphate backbone, and both use 4 different kinds of bases. And of course, they also differ in terms of their function. DNA is really the information storage molecule that to transcribe to produce RNA. And that's really its job. It has the instructions for making proteins. In order to make those proteins, you need RNA as the intermediate factor. So you have to make RNA in order to make proteins. You use DNA to make that RNA. And we use the same complementary base pairing to make all of this work. Coding RNA, another name for coding RNA is messenger RNA. mRNA. The N stands for messenger. And the message is how to make protein. The type of protein depends on the type of RNA, where that RNA came from in terms of gene. Non-coding RNA means that it doesn't give rise to a protein. So, messenger RNA, let's say instructed to a protein. The first of the non-coding RNA is tRNA. tRNA equals transfer. That's what the T is for. Transfers amino acids to site of protein synthesis. rRNA R stands for ribosome, ribosomal RNA. Makes protein. Synthesis of. And this is an important structural and enzymatic part of the ribosome itself. And the ribosomes are what make protein. The ribosomal RNA reads the instructions for the protein synthesis. tRNA carries the necessary amino acids required for protein synthesis to the ribosome. And sRNA, well, I simplified this. They have what's called microRNA, short interfering RNA. I'm just going to talk about it. I just don't remember. I'm just calling them all short RNA because that's what they all are. They're all RNA, relatively short to all the rest of this. They're little small sequences, and they're involved in gene regulation. You can think of it as turning genes on and off, but it's a little more complex than that. They actually can target messenger RNA to break down. They can block messenger RNA from being synthesized by the ribosome. So they're able to control the production of protein. That's a regulation of the gene product itself. They're so powerful when they were discovered, the first always used them to look at gene knockout models and gene reduction models. Alright, so that's the RNA overview. That's what RNA looks like compared to DNA. That's a comparison of coding RNA versus non-coding RNA. And I'm not done yet. Gene expression. I want to get to this part because this is at least a starting point for lab. Transcription. Transcription is just what this image shows. Transcription is using DNA to give us the instructions for making messenger RNA. This is how I make coding RNA. You notice that one of these strands is serving as a template. The top strand is top, the bottom strand is serving as a template. So when there's a E, there's an A. When there's a C, there's a G. When there's a G, there's a C. When there's a C, there's a G. They don't think any use for RNA. Why would they do that? I mean, this is exactly what you're going to see them use. So transcription is making an RNA copy from one of the strands of DNA, what we call coding. Translation is when we actually translate that RNA into a protein. We're going to do that in lab today. And RNA translation is based upon a 3-letter sequence. Every 3 letters is called a codon. And they give the instructions. This tells us what amino acid belongs here. In this case, serine. This tells us the amino acid aspartic acid needs to be here, needs to be linked together. And this tells us to put proline here and link that together. So it's literally instructions for a sequence of amino acids, which we all know is the primary structure of the protein, that then leads to secondary, tertiary, and eventually secondary structure. All right, go ahead and turn to your activities and we'll go to the lab.